We're beginning today by looking at the blueberry barrens of central Maine. And in this early spring view, you can see that it really is rather barren. The low bush blueberries that cover this area have yet to leaf out. And so you can see the ground and rocks and that sort of thing. But then if you notice way in the background, there is a line of trees because that's where the forest begins. So that raises today's issue. We are continuing on with our theme of the living world, but we're specifically going to be discussing the topic of diversity today. We'll start our discussion by looking at this little guy here. This is a kind of a sparrow called a grasshopper sparrow, which is a widespread species over much of central and western North America. But in northeastern North America, it has a rather limited distribution. One of the places that it does occur here in the northeast is on these blueberry barrens. But it doesn't occur everywhere. It occurs in that barrens habitat, but you would not find it in that adjacent woodland habitat. So this raises the issue of how do you go about determining what the diversity of an area is. And let's go back to some terms that we used in our previous show. We talked about the topic of species richness and species evenness being components of this measurement called diversity. Now, in our present discussion here, let's limit our consideration just to species richness, that portion of diversity. So if we're looking at the number of species that occur within an area, is that area one that includes just the habitat of the blueberry barrens, or does it also include that area of the forest behind it? What you want to consider the area under examination is going to influence how you go about determining the diversity, or in this case, species richness. Let's think about this a little bit further. We're looking here at an image of a place in central Connecticut called Cromwell Meadows. Now, Cromwell Meadows is a very extensive complex of wetlands, marshes primarily, but also some freshwater, shrub swamps, and floodplain habitat. Quite a complex of different kinds of wetland associated systems here, and all of them are present in this one photograph. And so we have to decide if we are going to talk about the diversity of this area. Are we talking about the diversity within a particular one of these habitats, or are we talking about the diversity of this whole region? Let's take a little bit different kind of look at this same spot using a different kind of technology. This is an image that's published in red, but it is actually an image that was taken using infrared light. And as it turns out, plant material reflects this light very nicely, and so it shows areas of vegetation quite clearly, much more clearly than we could see in the last image, which was exactly the same place as this. It makes the trees stand out in the center of the image very, very easily. We can detect them. So right in the center we have this line of trees. Water, by the way, absorbs infrared light, and so it appears black in the picture there. You can see the river and tributaries quite clearly as well. At the upper end of the image, we're looking at a shrub swamp, which again is more easily visible in this image. And then Marshlands, those areas that are covered with strictly herbaceous vegetation, those show up as kind of flat red areas. So much of the view here is of marshland, all along the river especially. And then you notice also that there are some white spots, and that is associated with human activity. I think that big white spot there is a gravel pit, sand and gravel or something like that, as I recall from being on the ground. So anyway, now we can talk a little bit more specifically about how this image relates to diversity. But let's do this by actually looking at an image on the ground of what this place really looks like as we would see it. We're looking here at that river that goes through this area, and on the right hand side we see marsh vegetation, and in the background we see some floodplain forest. So we see some of these different habitats within this one place. And so we could talk then about the diversity of species of plants and animals that live in just a marsh, or just a river, or just a floodplain forest. If we restrict our discussion to a particular kind of habitat, then we usually refer to that kind of measure of diversity as the area's alpha diversity within habitat diversity. If we considered these various habitats in this location, the between habitat compilation of all the species that are present, then we'd be talking about beta diversity. Now let's look at one larger scale than that. Let's look instead at the whole state of Connecticut, of which central Connecticut is simply one part. Now we're looking at an even larger scale with multiple ecosystems present. And so if we are looking at this larger scale picture of diversity, and again, let me emphasize that I'm speaking specifically about numbers of species. So we're really talking about species richness here. If we consider on this larger scale, now we are referring to what's called gamma diversity. So we could define them a bit more precisely like this. Alpha diversity referring to, again, within a habitat, or we might call it within a particular kind of a community, or even within a particular ecosystem. So we're looking at species within one thing. That's alpha diversity. If we are thinking of more than one ecosystem, and certainly in this photograph here we're looking at two ecosystems, the ocean versus the land, well, now we're looking at beta diversity. So we're comparing the number of species unique to each system in this kind of a situation. And our third kind of diversity is gamma diversity. And that is diversity of a region which contains multiple kinds of ecosystems. So like that view of southern New England. Now within the AP Environmental Science curriculum, there seems to be quite a bit of interest in looking at ways of calculating diversity, which is all rather amusing to me, because the idea of calculating diversity with information on both species richness and species evenness is an idea that really fell out of fashion among serious ecologists the better part of 40 years ago now. However, it is of some historical interest and remains of some practical interest to look at ways of calculating diversity. So let's talk about it a little bit anyway. One of the classic ways of incorporating both species richness and species evenness, and by that again I mean the number of individuals per species, the way of combining those things into one metric is by Shannon's index. It's often referred to as the Shannon-Weiner formula, which is incorrect. Shannon did work with an individual by the name of Weaver, and wrote a book about a mathematical topic called information theory that this index is based on. But this is really Shannon's formula. It makes use of the idea of taking a random walk across the countryside, and during that random walk, how many individuals of how many species do you encounter? So that's one way that was, back in the 1960s, a very popular way of computing species diversity for different areas. That kind of phenomenon was abandoned during the 1970s when people pointed out that by combining species richness and species evenness, you are actually losing information. It was more interesting to look at the components of diversity than the metric of diversity itself. By combining them, you couldn't any longer see what was going on with those two things, richness and evenness. One of the fatal flaws of calculating diversity indices is that two very different kinds of communities can produce an identical diversity value. You can have one community with many species, but most of the individuals in the community in just one or a few of those species, and you can have another community with few species, but with individuals rather evenly distributed among them. And both of those communities, even though they are really quite different in terms of their structure, can come up with the same diversity value. Now, despite all that, diversity indices still do have some utility in ecological research. I've used them, for example, as a means of quantifying the heterogeneity of plant communities in a single number, where I needed to compare plant communities to number of animal species present. Another area where they become useful is in looking at whether or not, when you've gone out and done a field study, whether you've sampled the system adequately. And I've done that as well. So, for example, in those marshes that we were looking at aerial images of earlier, I went in and did a whole series of surveys in that area, which was a good 500 acres plus of marshland, trying to figure out how many species of birds lived in those marshes. And because it was such a big area, and there's only one of me, and even though I went out a number of times, I wanted to know, had I really completely sampled that community? If I sampled that community adequately, could I then make valid comparisons to other marsh communities? So I needed to get some measure of sampling completeness. And another kind of diversity index, known as Simpson's index, allows you to do that. And there's also still one other kind of an index which is still used, called Fisher's Alpha, which actually has been around for quite a long time. That can be used similarly to look at sample size, whether or not you have an adequate sample size to make the kind of comparison that you want to make. So here are the formulas that are used for making these kinds of diversity calculations. And again, let's clarify, species richness means the sum of the species present, whereas species evenness is a measure of how many individuals are in each species. Now, there are a number of ways of calculating species evenness, but one of the simplest ways comes to us from very elementary statistics, doing something called calculating the coefficient of determination, which is the standard deviation of a series of observations divided by their average. And if you are unfamiliar with the term standard deviation, then I recommend that you look that up in a statistics text. It is a rather simple kind of a thing to calculate, but I don't want to go too deeply into all the mathematics of this in this program. So let's take a look at Shannon's index first. Shannon's index is computed by looking at the proportion of individuals in one species. If you look at the entire number of individuals present in a habitat, so let's say we find 1,000 individuals of all species present in a habitat, then what proportion of those are represented by one of the species? And then we multiply that by the natural log of that proportion. And then we do that for every species out there, and we sum all those numbers, and now we have Shannon's index. So it's an easy thing to calculate. You can do it on a calculator. It's a little easier to do on a spreadsheet, like an Excel spreadsheet, however. Simpson's index is also quite easy to calculate. In Simpson's formula, lowercase n sub I refers to the number of individuals in species I, and capital N refers to the total number of individuals in all the species. Now let's continue on with our theme of diversity, but let's change our focus a bit. We're looking here at a coastal salt marsh. So the plant communities there are really of one major type, and then these communities you can see are separated by creeks and bays and so on. But basically the vegetation area there is one kind of habitat. So we could again talk about the alpha diversity of the vegetated areas and the beta diversity of comparing the marsh itself with the adjacent water. But let's look at another image and see what happens when we get to the upland edge of the salt marsh. Here you can see the edge of the salt marsh on the left giving way to a border of grasses and shrubbery and small trees and things like that. So we are at the boundary between coastal marshland and upland here. And of course you might expect that different kinds of animals inhabit the salt marsh than inhabit that upland shrubby border. If we continue on back further inland from that shrubby border, eventually that border is going to give way to forest. Coastal forest to be sure, but forest. Coastal forest does tend to be rather different from interior forests because it is an area that is subjected to greater influence of winter storms in the northeast here, for example. And so it does have a bit more of an open character. In a lot of places it's also rather damp because of that moist air coming in off the ocean. And so you might get the development of lichens growing in big sheets from the branches of the trees. And you can actually see some of this in this photograph here. If you look carefully at the branches, you've got this beard moss lichen, also known as usnea, that hangs from the trees. For a long time, usnea here in the Northeast was largely absent because it's very sensitive to air pollution. So during the years of the coal-fired power plants and the steel industry of the Midwest, our usnea populations plummeted in much of the Northeast, especially southern New England, places like that. But as our air here has cleaned up, the usneas have come back. So actually this photograph here is of a place in coastal Rhode Island which has had a major resurgence of usnea populations in the last 10-15 years or so. So what I'm getting at here is this phenomenon, the edge effect, as it's often referred to, where one habitat gives way reasonably abruptly to another kind of a habitat. And in that kind of a situation, this boundary between habitat types is referred to as an ecotone. We're looking at still a different kind of an ecotone in the image behind this text here. You're looking at a farm field bordering a forest. That certainly is an abrupt boundary or ecotone. In the photographs we were looking at earlier, the boundary was not quite as abrupt. We had that marsh giving way to shrubs, giving way in turn to trees. But ecotone nonetheless. And historically, this has been an interesting area for wildlife managers because it brings together a larger variety of species. You have species from both habitats or multiple habitats converging in one geographic location, so the species richness of the area as a whole is high. And so wildlife managers years ago used to go out of their way to create ecotones to improve diversity of wildlife. But of course, as we've learned more, we've learned that that's actually rather a naive view of the way that ecosystems work. Despite this greater species richness at edges, many of the species that live at the edge do not do well. Their reproductive success can go down dramatically. So if we have forest species that are characteristically forest interior species, they do better reproductively in the interior of the forest than they do at the edge of the forest. And so the populations that we see at the edge of the forest are simply maintained by populations from the interior. But those edge populations actually don't produce enough young to maintain themselves. One of the reasons for that has to do with predator densities. Edges of forests can be places where you have a higher density of things like raccoons and foxes, coyotes. It could be an area where predation on nests or on young, one way or another, can be greater. And so consequently reproductive success declines. Researchers have also taken a look at food quality at the edge as opposed to the interior of habitats, and also discovered that, at least in some instances, the quality of the food declines at the edge and is superior for habitat specialists in the interior of their habitat. Now, on the other hand, there are species that specialize at living at the edge of environments. There are quite a few bird species, for example, that really prosper at that ecotone. There are species like the catbird, for example, that is very, very happy living at the edge of the forest, places where there's lots of shrubbery. Now, of course, the exact relationship between species richness and habitat boundaries or edges depends upon exactly what kinds of habitats we're talking about, what part of the world we're in. In other words, the whole picture is rather a complex one. And that is a theme that we see again and again in ecology. And that is, the more we understand about something, the more complex we realize it really is. Now let's discuss one more issue under the heading of diversity. But again, let's change our focus a bit. We're looking here at a picture of a marsh wren, which is one of the species that inhabits those wetlands that we were looking at earlier. But it doesn't inhabit all wetlands. And that brings up the issue of the species area effect. So let's consider this idea and see what I mean by it. My little friend here, the marsh wren, can live in very large marshes. And it can also live in reasonably small marshes, maybe 10 acres or so. But once the size of the marsh gets below about 10 acres, the marsh wren seems to pretty much always drop out, at least within southern New England. Now, it does prefer particular kinds of marsh vegetation. It prefers tall marsh plants with firm stems, and so we see this individual here in a stand of reeds. Marsh wrens do like reeds very much as places for building their nests. Now, it will also inhabit other kinds of marsh plant communities that have stiff stem plants, so it'll live in cattail marshes, it'll live in bulrush marshes, which all have tall stiff stem plants, but you don't tend to find it in salt marshes. The only place in salt marshes where you can find it is along the upland border, where you might have a border of reeds or cattails or something like that. So it doesn't like those lawn-like grasses that you find in salt marshes. So habitat does play a role in where it occurs, but all other things being equal, if we have marshes of varying sizes with ideal vegetation for the marsh wren, it still tends to drop out in the smaller marshes. So if we have 1 or 2 or 3 acres of reeds, very likely we will not find any marsh wrens there. Here's another species that exhibits a very similar kind of a phenomenon. This is the Virginia rail. It also lives in big marshes like that Cromwell Meadows marsh complex. It's rather common in there. And it will even inhabit marshes that are not all that big, 10 acres or so. But once the marshes start to get really small, a couple acres, then it tends to, again, to drop out. And so you can raise the question, well, why is that some of these species, even though you have a couple acres of habitat, they're not there? Some species are. Species like the red-winged blackbird are still in them, but other species like these two are no longer present. They're both little birds, and so they don't need a lot of land to live in. Their home ranges, the area that they occupy during the course of the day, is less than a couple acres. So one would think that at least one pair of these things could live there. But they tend not to. And so we can raise questions about why that is. We can say, do these species recognize these small habitats as being habitat for them? And the answer may well be no. Is there adequate food for them? One would think so, but unless one goes out and measures it, one can't be sure. Now, this is a question that's going to require some additional discussion. And so it's a question that we are not going to be able to finish answering today. I'm going to leave that for our next installment of our discussion of the living world.